Yep, we're very excited to have Dr. Robertson with us here tonight. He's quite accomplished, and we're going to have some other great speakers coming on board. So, like Robert said, get your dues paid so we can line everybody up. Uh, Albert is from Saskatoon, Saskatchewan. Gee, I don't know about you, but that just sounds far away and cold. But looking on the map, it is just north of Montana. So Dr. Robertson earned his PhD in 1982 from the University of Saskatchewan and the University of Alberta, Canada. He's published over 100 articles and has many patents. He runs 1,600 honey colonies and breeds over 2,000 queens each year. In 2004, they started the Saskatchewan Project, which I'm sure he's going to be talking about with other local queen breeders. And they're trying to breed the perfect queen that is both productive and gentle and resistant to disease and tolerant to mites. And with that, let's pass it on to Albert, and he can start the presentation.
All right. Well, thanks for having me. I look forward to presenting our work and going on from there. As you mentioned, we started the Saskatraz Project in 2004 with the objective to develop productive and gentle honeybees with tolerance to mites and brood diseases. And I think most people would agree that today our current honeybee health issues are mainly associated with Varroa infestation and the viruses it transmits, and there's a lot of interactions with the environment. Miticides themselves are toxic to honeybees. We have pesticide issues and nutrition, which is becoming more of a problem in some areas. So I just want to outline what I'm going to talk about today. I'm going to review our breeding program, talk some about biomarkers, which we've published on our website if anyone is interested in the details. I'm also going to discuss our Saskatraz Hybrid project with OHB in Orland, California. And towards the end, I want to look at combined miticide treatment experiments with selected and unselected stock. And at the end, we'll take a look at our commercial beekeeping operation in Saskatchewan at Meadow Ridge. So with that, we'll start on the details of the breeding program. We started a long time ago selecting for honey production, and that's a quantitative trait, which if colonies are good producers of honey, they usually are healthy, and most of those winter well, so they can build large populations in the spring. We also started doing Varroa resistance and suppression in 2004, work on that, and we always selected for brood diseases to colonies that were selected without shock brood or any signs of foul broods. And lately we started to work on viruses and nosema susceptibility. And the methods we use for Varroa selection, we use natural selection. We outcross, backcross, and use a lot of recurrent selection to try and increase the frequency of alleles associated with these traits. And the last few years we've extended our progeny analysis, and we do a lot of closed population mating and some instrumental insemination too, but more closed population mating. This is our original apiary, which was in a very remote area of Saskatchewan, a migratory bird flyway, and in about 2010 this yard's flooded. But the yard really represents this natural selection program and nothing ever come out of here. We just grafted to obtain progeny from the best stock and outcrossed that and backcrossed it within that apiary. To the right is the original team, my son Tom on the right, John Peterson, his son Eric, and my son-in-law Neil. They were the first to work on the project. This is a slide showing some of our Saskatraz lab and field crew actually. Dr. Shaw, he worked for us for a few years on mainly analysis of Nosema. In progeny analysis of Nosema, Mohammed Mostadran worked for several years on raising queens and progeny analysis in particular. Philip Griebel and Wayne Connor. Philip is innate immune specialist, and Wayne Connor did a lot of RT-PCR and PCR work for us over the years. On the right is Scott Napper. He's a specialist in the Kenome analysis. And on the bottom right is Dr. Shao Chu and one of our graduate students, Sanjay Chang, who worked on the gene expression work on microarray analysis. There's a graduate student, Sherry Wong, who's not in the picture, who followed up and also did quite a bit of work on gene expression and virus analysis. In the next slide we have here Dr. Abdullah Ibrahim, who worked for us in 2007, a specialist in hygienic behavior and Varroa biology. And of course, over the last 10 or more years, we have Antonio Manaz, who has taken over a lot of the queen rearing and queen production, and his brother Edmundo, standing beside some of our colonies. And on the right is Colton Rutherford, who has done a lot of work on data analysis and also works in the field gathering Varroa data and production data. The next slide, this shows us the breeding program's logistics. And there's a couple of main themes here. One is we started with a very diverse gene pool, and we do, the main theme is recurrent natural selection. But to start with, I started doing close population mating and selections in '92. We selected the best honey producers and good for wintering ability and brood diseases. And then in later, when Varroa became a problem in Canada, we imported Russian stock from Baton Rouge, Louisiana. They had a very good program on that. And I reconstructed some Russian stock, which was introduced into the program. And we collected stock from a lot of Canadian beekeepers who were in Manitoba and Saskatchewan who were raising their own stock. And in about 2004, we put all that together. And in Canada, the ones that were raising their own were wintering, so they've been selected a long time for wintering and for good productivity. In about 2012, I introduced the VSH trait into our population, with Bob Danka helping us with that, getting semen from colonies with high, from drones from colonies with high VSH activity. So we had a very diverse gene pool to start with, and we put them into the natural selection apiary with no treatments and followed their survival over time, selecting the best honey producers and ones that survived the longest. We back-crossed the best selections in that yard, so we were selecting for drones that were still viable in colonies with high Varroa populations. And we outcrossed those to maintain those lines and kept selecting for honey production, wintering, population growth, and recycling them through. And we distributed stock on a yearly basis since 2006. So just to flesh out some of the program there, one of the most important aspects now of our program is close population mating. So we have a number of breeding strains, some like SY26, very good for Varroa tolerance, and S96, very good for honey production. So we intercross at various yards that we have certain lines that have certain characteristics. We maintain, right now we just have 2 or 3 natural selection apiaries, but I've had up to 5. And of course we continue our economic trait selection for honey production, winter survival from about 1,500 colonies at 50 different yard sites or apiaries. So in the next slide I'm going to show what we do on a yearly basis in terms of natural selection. And you'll note on the y-axis on the left we have phoretic mite levels in mites per 100 bees, and on the right I have honey production in pounds. So we do alcohol washes for Varroa mite analysis, and we weigh all the supers for honey production. If you note, the brown bars are a honey production for each colony, and all the colonies are on the x-axis, and they are denoted as the strain and the year that they were placed in the natural selection apiary. So for example, SY26 was placed in 2014, and this is 2018 data, so she's still maintaining a very low mite level. In the spring we see not detectable. In the fall there is some. Honey production is good at about 225 pounds, but she's getting quite old. But also note that we have the mean mite per 100 bees on the red line running across the chart, and the honey production mean running across at the top in yellow. And we can see that the mean honey production, anything above that is eligible for selection, and anything below the Varroa mite mean is also eligible selection. And we see SL25, very good honey production, and it's a 2016 colony. So the older the colony, the longer they survive, the better they produce honey, and the lower they keep the mite levels are our selection criteria. So let's take a look at the next year, 2019, when the landscape changes quite a bit. We'll see the same coordinates here, but notice SY26, the mites are starting to take over. We see this colony has a slightly higher average than the mean, but she's also 60 months old now, and her honey production has dropped, but she's a good survivor. We take a look at a more recent colony in S96, placed in only 1 year. She still has excellent honey production and low Varroa levels. So that gives you an idea of how we do the analyses. This is for Varroa tolerance, but for honey production, we have about 3 or 4 close population mating yards. And the procedure here is that we select on an annual basis for honey production. So we weigh all the colonies, and anything that starts to decrease in production in that yard is removed, and we select from our other 50 apiaries for the best honey producers and wintering each year. There's a colony here called S28 that we placed in this apiary in 2012, and she is still, the colony is still surviving. She had pretty good honey production in 2019, 400 pounds. However, in 2020, she failed a bit, but come back. This colony I've seen this for years. I selected this colony in 2006, and I've seen on more than one occasion where this S28 strain will maintain the mother and the daughter, and they tend to supersede quite quickly, and it's a survival mechanism, in my opinion. We also have a colony that was introduced in 2013, S14, very consistent honey production over the years, and still maintaining a viable population. One that was another one, S96. These 96 strains tend to be very good for honey production and good heritability of honey production, and we see over the last 3 years very consistent. Also, we introduced a newer colony, SL25, which produced excellent yield of honey this year, and we are back crossing that to our natural selection yard as well as other to maintain that line. I just want to talk a little bit about Supersedure trial we did. Survival of honeybee colonies is a lot of times dependent on their ability to sense the changing environment in the colony. And if they can successfully supersede, that's a good trait in terms of maintaining survivability. So a trial that I did a few years ago, looking at the ability of different strains to supersede, and what I did was take one frame from each, or 3 frames from each colony, and put them in a separate 3-way nuke, and just observe in 10 days how many cells had successfully hatched, and recorded that as a Supersedure trial. And we see that all of the good colonies with good productivity and survival tend to be very efficient at superseding and requeening. These colonies do not swarm unless they're mismanaged. Usually they're quite good that way. So the next series of slides I'm going to show you is about progeny analysis. This is something that we worked on for many years, and just to explain how we do this, we'll take a breeder that we select, then we line out the daughters. In this case, it's an S113 breeder with 8 daughters, and we measure the mite population in their infestation in the fall before they go into winter, and again in the spring when they come out of winter. And we look for colonies that in the spring show very little, if any, phoretic mites. This is a good way of determining whether the bees can actively groom and get rid of the varroa population during the winter. So we would select S113-2, -5, and -8 for further analysis or breeding for maternal and paternal colonies. The next analysis or type we've done is nosema analysis, but where we measure the number of spores at different times of the year, December, January, February, and look at the level of spores. And this is a particular S88 colony, which we did 8 daughters, and of course we tried to look for ones with the lowest spore count. However, I stopped doing this. It's too laborious. Nosema is something very difficult to breed for, so we just quit treating any of our colonies for quite a few years now. The ones that are susceptible eliminate themselves from the population. We also did a lot of analysis earlier on. This is quite laborious, the mite biting analysis, and you'll see on the y-axis the mean percent damaged varroa. This is a lot of microscope work, and what we found is that colonies that were most showed the highest mite damage in the analysis were the longer survivors. So 30, 28, and 34 showed good activity and were the best survivors. Colonies the 24 and 06 were very susceptible mites with less activity. I have a video now I'd like to show on this. I placed a pupa on the brood with a varroa mite on it. Then it takes a bit of patience to do this, but I've seen S28, for example, be very aggressive against a mite once they detect it. So hopefully you can see.
She got away from it right away.
You see the pupa. There's a bee now.
Oh, that one's got it.
It's got it. It's chewing it.
I got it.
Yeah.
I got it.
She's chewing it.
So that's what I like to do when I'm selecting in California with the breeder colonies there. The next slide, I'm going to show you a new method of assaying for varroa-sensitive hygiene. We've just initiated this study with Dr. Kyra Wagner at the University of North Carolina. And we obtained some of the compound. It's a cuticular hydrocarbon that has been synthesized, which is produced in pupa that have been stressed by varroa. So what we did here is on the left, you see the control with the green dots where these cells are all closed. We sprayed this cuticular hydrocarbon on there and put the frame back in the colony. And 2 hours later, on the right, you see the results. Notice that 84% of the brood cells have been started to or progressively have been opened. To me, that's amazing, because in 2 hours we can now determine whether or not, or measure the VSH activity of our colony. So we're actively pursuing this to do progeny analysis for VSH activity using this UBO, or unhealthy brood odor assay. Now I'd like to move into some of the work we've done in the molecular field. I don't know how many of you are familiar with molecular biology, but we'll take a look at what we've done. This is, if you wish to look into this in more detail, you can find the publications on our website. But I started working with microsatellites as soon as we had Russian stock. We managed to identify about 20 informative markers or microsatellites for distinguishing between Russian, Canadian, and New Zealand and American bees. So we were happy with that. We couldn't identify any markers, though. It was more or less for identifying different bee populations. Well, later on we looked at doing some transcriptional analysis. Now it's RNA-seq. At that time it was microarrays. I had 2 graduate students work on that. I've done some proteomics, which I'm not going to talk about at all today. And Kenome analysis I will describe, because I really like this analysis, and I think it's potentially good for having markers for varroa tolerance. So to start with, I'm going to describe our work with transcriptional analysis. We screened 13,000 contigs sequences for expression in both a susceptible and tolerant line. They were extreme phenotypes. G4 was susceptible and S88 tolerant. And we looked at how they responded in the infected and uninfected pupa. So you'll see a column S88 infected plus over G4 plus, and I've highlighted in red the most differentially expressed, and we see 7-fold almost increase in apolipoprotein D, and 3.5 to 4 increase in esterase E4, and cytochrome P450 was also increased by 4-fold. All of these transcripts, when transcribed into proteins, are involved in stress tolerance and detoxification mechanisms. So that's quite important for the survival of the colony in the presence of varroa from this data. So survivor colonies, just to cap a bit on that, why do some colonies survive for extended periods? In natural selection apiaries, we've had S88 survive 58 months, which I just showed you some molecular data on. And SY26 was the longest survivor at 70 months. Now these colonies, this is colony survival, and I know with SY26 it superseded at least 2 or 3 times. And SA80, I never did see evidence for that, but it's possible. So let's just review what we've seen correlated with survival. We see grooming behavior, colonies that are aggressive towards mites. VSH activity is important. SA88 showed very good varroa sensitive hygiene, so that helps keep varroa populations low. Supersedure rates, you see that if a colony is able to supersede successfully, it helps to maintain the colony survival time. And stress resistance, colonies that show higher levels of detoxification factors for pesticides, miticides, and environmental stressors also show better survival. I'm amazed this apolipoprotein D is really an important component. It travels in the hemolymph and it is a clotting mechanism in the hemolymph. It serves as that. So if varroa are making puncture wounds in the pupa, they are able to heal easier. So that's quite a good stress protein. Better foraging activity. If we're selecting for honey production, we're selecting for better nutrition and pollen gathering activity. Virus immunity is another thing that we're looking into now, and we've got a collaborative study with the USDA Baton Rouge in Baton Rouge, and they're assessing some of our strains for in vivo analysis, infecting them with deformed wing virus. But I have some data on that, which I'll show you next here. This is an experiment where we looked at infected and uninfected pupa, and what we did was isolate the RNA from those and assay for deformed wing virus infection levels in the head tissue. On the y-axis is the CT value of deformed wing. So what that means is we're measuring the number of viral copies, and the higher the value, the lower the virus infection. So if you look at SY26, one of our long-term survivors, in S23A, we see that the infected state of the pupa and the uninfected state have nearly the same levels of deformed wing virus. So this implies that these colonies are able to actually perhaps decrease the replication of these viruses. However, a susceptible colony, when infected, shows very high levels of the virus in the head tissue, and this could compromise its survival. So we're interested in doing more work on this. This is another slide showing a replicate of the deformed wing virus in head tissue with and without mites on the left panel. And again, the CT values, the higher the value, the lower the deformed wing copy number. We see that that in Y26 again, the virus level is still lower with the mite infection. It's still not that high. So it's where there's a S14, an intermediate phenotype, has a little higher value, but the susceptible has quite a high value of deformed wing virus in the pupa. The next panel, the right panel, shows us a rather unique experiment which we did in vivo here. This was where we applied some miticides to the pupa in different strains of bees. So the CT values again are the same as in the other charts. The higher, the lower the deformed wing virus number. And you'll see in the controls is red, apivar is the orange triangle, apistan the yellow square, and thymovar is the green diamond. In the tolerant colony, we see that the virus levels are about the same. They're not significantly different. Using the LSD, at least significant difference, they're the same. Whereas in S14, which has not quite as tolerant as Y26, we see a significant difference. There's some increased deformed wing virus in the apistan and apivar treatments, and only a small change with thymovar. But in the susceptible, there is quite a large difference. The apivar deformed wing virus levels are very high in the susceptible colony, and also in the apistan treated, but less with thymovar. So this is something that we're pursuing now. We're doing more tests on that to see if we can repeat this in full colonies with the adult bees. The next section here deals with Kenome analysis. Now, kinase activity is really important in response to any type of stress in the environment, because its messages, it starts throws the switch for a lot of metabolic pathways, and then with bees, as soon as they're challenged with varroa, you see the pupa there, it starts a whole cascade of reactions within the organism. But the picture here shows a Kenome peptide array, and I just want to show you, we were quite delighted with this because it worked very well. Each one of the spots on this array is a 15-mer peptide, and the peptide has a phosphorylation site in the middle, and when it is phosphorylated, it lights up, and it can be measured according to the intensity to the degree of phosphorylation. I've just shown you these are printed in triplicate, giving us 9 technical repeats, but I want to show you the reproducibility of this technique. You see here in this replicate, here, very reproducible. So all of these, that makes it very worthwhile in terms of screening our colonies for differences in phosphorylation events between infected and uninfected pupa in tolerant and susceptible lines. So what we found over our studies over several years, that we were able to identify 19 differentially phosphorylated peptides between pupa collected from varroa susceptible and tolerant colonies. And our bioinformatics, of course, picked peptides for analysis that were involved with activating and inactivating innate immune pathways, metabolism, stress responses as well. So this allows us then to screen colonies for their degree of tolerance. So in the next panel here, the chart, I'm showing the effects of, this is a principal component analysis of the results from the Kenome arrays. We see that we're able to discriminate between susceptible and tolerant colonies. The red are the tolerant, and the susceptible are green. There's one tolerant bee that didn't fit with the rest, so it wasn't expressing the same kinase activity in that population at the top left of the panel. If you look at the susceptible colony, you see that they're also separated on the basis of whether they're varroa infected or not infected. So this tells us, and the interpretation of the Kenome data indicates that the susceptible colonies, when they're varroa infected, their actually innate immune system is down-regulated. So the varroa is having some effect on the innate immune system of the susceptible colony. So this is quite interesting for further investigation. These bees then become more susceptible to virus infections. This is the last analysis that we did, and with the Kenome, and is published in Nature. It'll be on our website if you are interested in looking into it in more detail. But to explain this chart, on the left side is a colony survival in months, and the white bars indicate that, the colony survival time. And honey production is in the cross-hatched orange here, and the mean phoretic mite levels are in the cross-hatched red, and the mite levels are on the right side of the chart here. And we also have axes for the biomarker susceptibility score, which is calculated from the differences between the kinase activity of the susceptible and tolerant. But one thing that I want you to notice here is that the biomarker scores in the long-time survivors here are quite low, whereas in the colonies that are susceptible, these purple dots indicate their susceptibility. So the difference is quite extreme between highly susceptible and tolerant. So we can print arrays and actually screen them and predict whether or not they're going to be susceptible or tolerant. We need to do more work on this, but this is quite exciting, I think, going forward to help with our selections. Now, moving to a different section, I have worked with California queen breeders. I started working with David Powell and Ray Oliveris about 10 years ago. And this was a request made by Canadian beekeepers because they wanted us to produce queens with some relation to our Canadian stock in California so they could receive them early in April and May for their colonies. So that's the reason it was started, was to commercialize our stock. So every year we send about 150 or more breeder queens to Orland, which have been selected as I've showed you over the years. And I like to go down there and reselect them to pick the best of the set. This also serves to help with the hybrid queens, since we can produce a large number of them. The drones from these colonies carry the alleles of the breeder queens, so they help to distribute or increase the frequency of alleles associated with economic traits in the commercial populations. We now have stock in many different places, Middle East, Ukraine, South Korea, and so on, and they have done quite well over in those countries. I was surprised at that, but they seem to produce good honey, and honey is valuable in those countries. We're working on trying to get our stock into Hawaii now, and Australia is interested as well. This is the team that I work with in Orland. You see Ray in the center here, his son Joss, Jose Gomez, who heads up the queen rearing team here. These ladies do look after the cell builders and do the grafting, and they work quite well. They run an amazing number of cell builders. I'm quite pleased. They do excellent work. Just to give you an idea of the differences in our climates, in Saskatoon we actually had -50 with wind chill not long ago. So we have a very wide temperature range, extreme climate. I'm lucky if I can raise queens 3 months of the year. In California they got 5 to 6 months. It's much better. So that's one of the reasons why we started this project. So just to compare the two areas, in Saskatchewan I focus on production of breeder queens by the methods that I've described: recurrent selection and close population mating procedures. We have good location for selection in our extreme climate. We can produce up to 2,000 queens a year, which we send to California. So the hybrid project is focused on large-scale commercial production, and I haven't been down in the last year, but hopefully COVID settles down and I can get down there again this spring. We got and get reselecting the breeders again. I'm amazed at how lovely the colonies are when they come out of the almonds. They're full of nectar and honey and pollen and booming, with lots of mature drones, so they're ideal for mating. Here's some of the mating locations in California. They use styrofoam mating nukes, which is different from what we use. Just on the left is they mark all the areas with what queen lines are there. This on the right is a drone shot of some mating areas in or around the Orland area. This one too shows an aerial picture of the mating. You can see the drone mother colonies, some of them here. And in Saskatchewan it's a little different, but here this is a close-up of how they mark. So these would be, in these mating nukes would be cells from our SY26 breeders. So they know which ones they're catching and they keep track of the different lines, which is great because that takes a lot of effort actually. This is in our neck of the woods. This particular yard is the Purdy yard where I showed you we'd selected for honey production. We run 3-way mating nukes in standard supers sitting on top of some decommissioned boxes. This is our one of our natural selection yards where we have them. These are close populations, so they're isolated and close to the colonies that for their drone source. So just to give you an idea of what I do in California when I get down there, I do these colonies that I'm selecting already have been selected as breeders in Saskatchewan, but I go through them again and try and reselect the best of the lot. So I take a look at temperature or temperament, and that I do as I work them. If I get stung too many times, that's a black mark. I like to watch them on the comb. I like calm bees and how they're vigorously dancing if they got pollen to communicate things. Low temperature flight, the earlier they get up in the morning and go to work, the better producers they usually are. Queen retinue is something that I've done for a number of years. That tells me how well the queen is mated, and also tells me usually is correlated with the productivity of that colony. Swarming, supersede, pollen storage, I look at that, propolis production. Of course, brood pattern, worker uniformity, queen color and markings, and I do in situ varroa assays. so that I can tell whether or not there's some activity against the varroa. The Bee Informed Partnership has looked at our colonies several times over the years, and I like that because they do the freeze kill test, which I don't have time for, and they do all the other things, temperament, pollen, and so on, and they measure varroa infestation as well by alcohol washes, so that's nice to have independent assessment. Here's an example of when I'm working a colony. The first thing I look at is I find the queen, I take pictures of her, and when I look at the queen, one of the first things I look at is whether there are 3 distinct rings below the wings. That tells me this queen is well mated and likely laying well. If not, it could be poorly mated or a virgin, actually. This hygienic behavior was done by the tech team, but you see nice brood pattern, uniform bees, calm on the comb, etc. So we'll take a look at what I do for a varroa assay. When I'm working in a colony, I look to find a drone brood. If I find drone brood, I lift it and look for varroa. I like to do about 50, so it gives me an idea of the varroa in the colony. We lift the drone brood, and then if I find a varroa on a pupa, I record how many varroa I find, but I also do an assay with it. And this one is one that I do for grooming behavior. When I put this pupa down with the varroa actually hopped onto a bee. They're really fast. They can jump very quickly and land on a bee, but we caught that. There's the mite right there. And she's vigorously grooming that. She's grooming that. But this is an example of auto grooming. I didn't see in this particular assay any allo grooming. It was mostly auto, but she finally gets it off. So that's something you want to watch for. Take some patience, but is of interest. Here's an example of a very nice queen retinue on an S96. You notice how the bees are all very attentive to the queen and they follow her around. So that's a good sign that that's a very good queen. This is a video of a queen laying, and it's to me it was quite exciting to see how quickly this queen laid and how the retinue changed.
The bees are. Look at the retinue building. See?
Yeah.
She's busy. Oh, there she's laying. Keep on, girl. 1,000 a day. How many seconds did it take her to lay?
Let's find
Oh,
out.
she's doing another one right now. Oh, wow. She's laying up a storm, eh? And the bees are right around her.
That sun's out there. They're ready to go.
Look at that. The bees are really... I don't know if they're encouraging her or what they're doing.
They're just surrounding her though. She's got her head in that cell.
And she's looking at another one, but she laid twice.
She goes.
Yeah, she's out now. She's in another one.
Yeah.
Right away. Wow. Look at that. Another one.
So that gives you an idea of what I try to do in California. I wanted to, after several years, this is in 2016, this data I'm showing you here, and it's an assessment of the Saskatraz Hybrid performance in Saskatchewan. Because, you know, there's this idea that bees produced in the same locality are better than ones produced elsewhere, but in this particular assay we didn't find that. This graph shows you on the left is the mite level, and on the right is the honey production in pounds. We have to ignore the first mite analysis here in blue because it wasn't synced with the fall, with the green ones, the mite before fall treatment. So the thing to point out, I want to point out here, is that the honey production in the hybrids on the X, you see the strain designation S14 in Saskatchewan, S14 in California. Hybrid honey production is similar, if not a little better than the hybrid. We see that for most cases, the honey production is equivalent or not much different. This red line here is the Saskatchewan honey production on the provincial data, which would be average production for the province, and you see pretty well all of the lines are better than the average production, and there's no real difference between the hybrids and the queens raised in Saskatchewan. In 2018, I was invited to UAE. I sent some breeding stock over there, and this is a picture of the Saskatraz apiary in Hatta, which is near Oman, and this apiary wasn't far from the Strait of Oman in the Persian Gulf. But our bees are under this net here. They try to cool them by putting shade cloths up, and the breeders are in here. I don't really like their hives. It seems to me they don't help with the heat. And this is an Egyptian beekeeper that was looking after our bees there. And this was the owner of the territory, Sheikh Mana. What a nice fellow. But what I did, I went through a number of colonies there. Quite a few actually from Abu Dhabi to Oman. And the Egyptian bees that they keep are very mean. Their temperaments were bad. But this was a nice colony. I like this. This is Apis mellifera yemenitica. This colony had very nice brood pattern, and were calm actually. I don't know about their honey production, but they seem to have good pollen placement. This is a Saskatraz drone that is visiting Mrs. Yemenitica here. One thing that stood right out is the different sizes. Our bees quite a bit bigger than the Yemenitica, but I was looking forward to crosses with our bees, but that hasn't happened yet. The miticide treatments in stock selected for varroa tolerance. We can, to summarize what I've talked about actually, we can select survivor colonies which show excellent tolerance to varroa, but it is difficult to stabilize the trait in the progeny. Like our SY26 has likely the best heritability at 15%. Of the daughters are equivalent to the mother. And that's disappointing and difficult to stabilize the phenotype. So in commercial production apiaries, we still require miticide treatment protocols. So we have found that stock selected for varroa tolerance improves the efficacy of the miticide treatments. In addition, in our experiments, we found that combined treatments show better efficacy as well. I'm going to show you some of that data. But we've also found some miticide treatments may increase virus levels. And I showed you the in vitro stuff that we did, or in vivo, with Amitraz and with Apivar and Apistan. So I'll show you a comparison of data from Saskatraz stock and colonies not selected for varroa tolerance with Apivar, and also some experiments on combined miticide treatments. We did 10 different trials here, and we picked 3 combined treatments that in preliminary analysis looked promising with a higher replication. So that's what I'll describe as well. So in the first top panel, we see mites again on the y-axis, and the before treatment with apivar in the fall, and then in the spring on the right side. In the red bars represent Saskatraz colonies. There was quite a few colonies here. I just forgot how many, but there was a significant number. And the other two bars are unselected, the yellow and the brown. You can see there's significantly fewer varroa in the spring in the Saskatraz colonies than the unselected. And one of the most notable things was their survivorship. The survival in Saskatraz colonies was 85%. It was 50 to 60% in the unselected. So there's quite a difference in the efficacy using selected as opposed to unselected colonies. Here's the miticide trial that I was talking about, that we narrowed it down to looking at apivar and oxalic, apivar thymol, and apivar thymol and oxalic together. There was 15 colonies per treatment, so the replication was good. And we started out in the 4 different treatments with similar levels of mite, phoretic mite levels. So the first treatment we looked at was spring, mites per 100 bees in 2016 before treatment. You see in the blue here. And then in the fall, we have, of 2016, there was no fall treatment. So what we found later in the spring of 2017, before treatment, there is some mite presence, not high but significant, in the spring, and in the fall, before fall treatment, the mite level has grown to 4 in apivar. In the apivar oxalic, it's less in the fall, and the apivar thymol, the mite levels are staying quite low, and in the thymol plus oxalic with apivar, they are also low. However, in the spring of 2018, before treatment, the mite levels are up in the apivar group. In the apivar oxalic, they're quite low and not detectable in apivar plus thymol and apivar plus thymol plus oxalic. But the percent survival is also significant. So this trial ran 34 months. You can see that the survival is 27% in the apivar group, whereas the combined trials are 40 to 47%. So that's quite a difference. So we're seeing better efficacy with the combined treatments. So just to summarize and talk about work we have in progress, we can select Saskatraz families with good honey production, wintering, and varroa tolerance, but difficult to balance the phenotypes. Varroa tolerance is variable in the progeny just because the nature of bee genetics. There's a lot of outcrossing occurs, high recombination rates, and multiple drone matings. So our focus is aimed at stabilizing this mite tolerance using extensive progeny analysis with the marker-assisted selections that I've described and the new UBO assay to speed up our selection process so we can combine the best maternal lines with the best paternal lines. We also got a collaboration with the USDA and Baton Rouge, where we're looking at virus susceptibility in our strains. We have quite a few down there, and one paper is already come out, is being published in Frontiers in Insects. We're also studying the effects of miticide treatments on colony virus infections and what impact that might have on honeybee health and varroa population growth. We've got some funding from Project APIS-M to do that, and we're collaborating with Dr. Declan Schroeder in Minnesota and Patricia Wolf in Alberta. So I'd like to move on now to give you an idea of our commercial beekeeping operation. Now that we've gone through the breeding program and what we're doing, just to give you an idea of what we do for a living here, we run approximately 1,500 colonies for honey production. We're spread out quite far. We have 50 apiaries in over a 70-mile radius, although that varies likely between 50 and 100, depending on how much isolation we need. So several apiaries are isolated for close population mating and stock maintenance, so we're able to do that. We winter over 2,000 colonies each year. We produce about 700 to 1,000 nucs for replacement stock and for sale. The last several years we've been using 3-way nucleus colonies for requeening and double 3-ways for queen banking, and we're wintering those as well. So, and we produce over 2,000 queens every year. So in our area, honey production is our bread and butter. So I'll just show you some pictures of peak honey production. The top left panel, you'll see this was an exceptionally good production from this yard, and the bees were very populous, and the honey had been stripped. They were right full, and they will be full again in a week after this to 10 days. We get very long days and lots of canola bloom and heavy flows. All we need is a bee population and healthy bees. As below another yard, that's SY26 yard, and an exceptional colony that I selected. It had likely 250 pounds of honey on it when we first stripped it, and I grafted from that one. This is in the fall, wintering. We winter a lot of double 5 nucs because I make we make sometimes too many late crosses, and this is an example of how we put them in our wintering shed. It was actually used for a potato storage at one time, and we feed them before we put them out, but sometimes in the winter because they get quite thirsty, it's a dilute syrup solution. We use about a 3-to-1 dilution with water. Just one thing that's important, we do spend a lot of time labeling our colonies, and just to give you an example of, I'd like to see when we walk up to a colony, this, we know it's a Y26. We know where it was mated here. We know who the mother is, that's 2014 colony, and we know where it's at. And we use a smaller ID for sampling ID. So all of the colonies are labeled like that, and if we leave the cages labeled inside, because sometimes the labels get pulled off. The next I'm going to show you of what we do sometimes indoors. This is under red light, and it's a video showing how we treat with oxalic liquid dribble before the bees are moved out, and when the bee population, or there's very little sealed brood in the hive. So we use the trickle method. I use 50 ml syringes because they work well for a while. Once they wear out, then we just use a new one. I have tried the fancy dribblers, but I gave up on them. We put pollen on them, and they're being fed, so they'll be moved out when the weather's right and they're ready to go. They start laying quite readily, although we found too that if we feed them before we put them out, they're not so crazy to fly, and we don't have so many non-returning flights, because I think if they're very thirsty when they go out, they tend to just want to fly to find water. So these will likely be moved out shortly. I'm the smoker man with the light, and I noticed I left my suit unzipped, and I found out later. This is what it looks in the daylight. These are 5 nukes. They're ready to go out. They've been treated and got some pollen, fresh pollen, and they are look very nice, uniform and full of honey. This is some indoor wintering with 3-ways. You'll notice stacked on pallets. Each one of those doubles have what we call double 3-ways. So they have 3 frames in the top, 3 in the bottom, and 3 sections with a queen in each section. And I'll show you a video here of transferring these. They are amazing. Some of them come through very nice and strong. You'll notice that there's no visible or very little visible cap brood. So they have been treated with oxalic liquid to remove any phoretic mites, and then are being transferred into these are strong enough they'll end up being 5s. But it's a little bit of a pain because you have to find the queen to put in each nuke, and there's a lot of drift because of the number of bees flying around, and we have to try and shake the bees into the proper nuke. But they're economical to do because you got a lot of laying queens in a small space. So we also tried this year 6 queens per double. So I've got 3 frames of bees with a queen. So in a double standard super, we have 6 queens laying. And I have failed so many times at trying to indoor winter queen banks. They usually die on me in February that I've tried to do it using 3 frames of bees from our mating nukes. And these are mating nukes that we adapt to that. So right here, we also have the option with these in our wintering shed. We find weaker ones, we put them in 3-ways. We double whatever tends to be weaker, we put them in a 3-way. And these have been transferred but not treated to 3-ways. You'll see we got a cage in there to identify the queen, and we can treat them and keep them indoors and feed them until the weather is better to put them outside. So that's a nice option to have, and once they get the right conditions, they take off pretty quick. Our outdoor wintering, we winter, like I said, about 1,500 outdoors. We have the spring treatment again is the Apivar that we use in oxalic with 3.2 grams per 100 mils of 50% sucrose dribbled on. On the left is 2021, on the right was 2020, I believe. So sometimes we have to do it in the snowbanks because we try to do it at the end of March, early April. So I'll just show you what we do on the outdoor. We take the lids off in the top pack and inspect the bees, lift the lid off, smoke them a bit. We like them to look like that, wall to wall, and then we dribble oxalic liquid and we put in 2 strips of Apivar, and we sample them, and then later on they're fed. And we leave loose pollen in the yard in the pollen feeder you can see in the right picture. So here's just the same thing again. We sample off the lids. I don't like to disturb the colony very much at this time of year because breaking the seal on the lid is bad enough. And then we put the pails on and feed them, and that's basically how our spring goes. So the last slide here I have really is on the some 5 years of results doing what I just showed you in the top panel. mites per 100 bees before and after spring treatment, April to August. You see in April, in 2016, we treated with Apistan in the fall, and we had quite a high level of mites in the spring. Then we used Apivar and oxalic, and they were down by mid-August, quite low. 2017, we're just use the combined treatments, and you see each year it tends to get better. And I think part of that's due to better varroa tolerance in the bees, because the synergistic effect of bees selected for tolerance and with the combined treatment. I mean, these are beautiful 95% alive in the Apivar thymol, but just beautiful in the spring. This is another panel here showing similar thing with thymol. Thymol works well too, but the combined treatment works better. We see here that the mite levels are very, very low in the spring after fall treatment with Apivar and thymol. So the combined effects of combined treatments and varroa tolerant stock is giving us very good results. So to end, I've just acknowledged some of our collaborators here. We support our breeding program by stock sales, and we've had contributions from our collaborating beekeepers in Saskatchewan. For molecular work, we've applied for public funds, and we have had some funding from APSM this year. We are collaborating with USDA. They've been very helpful with our breeding program over the years, and some of the other people involved. So with that, I can stop and ask any questions that you might have.